So today we will have a lab video wherein we demonstrate this very interesting software tool called Cytoscape. So I will introduce you to Cytoscape as well as the string database, which is a very good repository of different protein-protein functional association networks, and I will also talk you through how you load and visualize networks using Cytoscape, and a very interesting plugin for doing a lot of network analysis in terms of identifying centrality measures or even visualization, which is known as NetworkAnalyzer. So today let us look at Cytoscape, which is a very powerful tool for doing network analysis. We previously studied how we can do network analysis with MATLAB BGL, right. That is a little programmatic, and there is also this other tool known as this other Python package called NetworkX, which is very powerful for doing network analysis. Today let us see how we can do it more simply through a graphical user interface. This is Cytoscape, and Cytoscape can actually be used to prepare really beautiful illustrations and so on. So Cytoscape is available from cytoscape.org, and it has many features. We will probably just try to look at a few simple features. It has several interesting useful plugins and so on, which are known as apps. So for example, there is a string app, which is probably the most useful because you can directly get string networks into Cytoscape. Let us maybe try installing one of those. So you can directly fetch proteins from the string database and start expanding the network and so on. But more than using the string app or any of those things, I think the best way to go about this would be downloading network information directly from the string. So let us go to stringdb.org. So string has information on 2031 organisms, about 9.6 million proteins, and about 1.3 billion interactions in all. Right. So we can go to download. The good part about string is it allows downloads on a per organism basis. So you can just pick an organism of interest, let us say Salmonella typhimurium LT2. So all organisms are basically identified by their tax ID, the taxonomy ID, which is something like 99287 for Salmonella and 511145 for E. coli if you want. So you can pick any organism of interest. So let us see how this file looks like. So this will be quite a large file. So it has 3 columns: organism ID dot protein ID space organism ID dot protein ID space score of interaction. Do you remember how the string score is calculated? So string has information coming from several channels. Right. So just let us just go back to string. So this is the first protein from Salmonella, which is THRL or whatever it is. So you can do a lot of interesting analysis right off of the string database itself, but I think we would like to do it computationally offline, right. So this even tells you if there is a structure available and so on. Can you see that there is a structure that is there, right. But what is interesting for us is this. Can you see the multicolored lines? Each of those lines represent a different channel of interaction coming from curated databases, experimentally determined, gene neighborhood, gene fusions, gene co-occurrence, text mining, co-expression, homology and so on. So you have multiple evidences that are available for every interaction, and these are finally integrated. So how are these integrated? So string has a nice way of integrating this database information. Let us say your scores are, let us say you have a protein P1 and a protein P2 and the interaction score is some 0.35, right. And for the same P1, P2 you have another interaction score of 0.45, this comes from phylogeny, this comes from say text mining and something of this sort. How do you integrate these scores?
Take average.
You could take average, but the idea string uses is a little better. What it does is, if this is, you can consider this as a probability of interaction, right? How do you combine these probabilities? So it depends upon what you want to ask. So if this is the probability that P1 and P2 interact based on channel 1, this is the probability that P1 and P2 interact based on channel 2, and this is the probability that P1 and P2 interact based on channel 3. What is the probability that P1 and P2 interact? So it's going to be 1 minus 0.35, 1 minus 0.45 into 1 minus 0.86. Right. So basically the final interaction score is going to be 1 minus does that make sense? So what is the probability that they do not interact at all? The probability that they do not interact according to this, according to this, according to this will be this number, product of these probabilities. 1 minus of that is the probability that they interact. So this is how you combine, string combines these scores up. So it's quite useful. So this is very useful if you choose to take the string, there is one more file here actually. So there's this file with links detailed. Right. So this will have all the channel specific scores. So if you take all the channel specific scores and you want to discard a couple of channels and reweight the scores, now you know how to do it. And in string there is like a small one nuance is that all scores are given in 3 digits, right. So it will be written as 0.356 or actually just 356, and there are a few cutoffs for confidence. So 400 is medium confidence, 700 is high confidence. You can also use 900 if you want. I like to use 900 to start off with because it will give you a very sparse network that can be more easily analyzed. But if you are going to do it computationally, you can do obviously any confidence that you want. So for using Cytoscape, I prefer something like 900. So let's see how we go about doing that. So let's import a network into Cytoscape. So you already have a filtered network which has only the links above 990. Maybe that's too much. Let's start with 950. And I can assign something to each column. So this I can say this is the source node, the first column. The second node is the second column is the target node, and the third column is basically an edge attribute. You may have it to be disconnected. Let's just see how it looks. We can also lay it out for you, and it usually has nice layouts. Yeah, so you see there are so many components clearly. So let's look at the style. Maybe there are different layout styles you can have. I think you can't even see it. It's already not. It's too dead. Too many nodes. But if you start zooming in, so this is what the network looks like. So you can see like there are so many separate components and so on. Let's maybe look at 990.
So even if you look at 990, the number of nodes are going to be same. Just the edges will not be.
No, so the orphan nodes will be removed.
So what were these? What were?
So these are orphan clusters. Right? They are small clusters with If you zoom in, it will show you also the different features that you have and so on, like the any node features, so you can show node labels and whatever else that you want. The most useful aspect is there is a network analyzer. You can just say analyze network, treat it as undirected in this case, okay, and it just computes a dozen properties, right. You can look at the node degree distribution, looks reasonably power law, clustering coefficient distribution, number of shared neighbors, shortest path lengths, topological coefficients, neighborhood connectivity, betweenness centrality, closeness centrality, other measures like stress centrality, whatever, and so on. The other useful thing is you can just go and say directly visualize parameters, right, and map node size to degree, right, and to say apply. The zoom can sometimes play up a little. Yeah, so you can see some nodes that are larger here. See, you have like such a tight cluster here. You have a lot of biggish nodes here. You can even just move out a single node, or you want to just delete this node, you can just delete it. You can even select the first neighbors and so this is how you go about studying perturbations, right. I just removed a node and all its edges, and now you can recalculate. You can recalculate properties and so on. But obviously you can imagine it's going to be a lot harder doing it with the GUI and so on, which is why I tried to focus on MATLAB BGL earlier. But this is another nice way. If you have a smallish network and you want to visually try to understand a few things, this is a great way to go about it. You can directly visualize betweenness centrality or closeness centrality or any of those measures and see how it affects.
There is built-in for perturbation.
Built-in app for perturbation or something? That might be something. I'm not aware of it. There's just so many different things. Something for motive discovery. Yeah, see, and you have nice things actually. So you can even decide how you're going to visualize it, right? So what I showed you here was I did some auto visualization using the NetworkAnalyzer toolbox itself, right, plugin. So what you can actually do is you can pick what you want to visualize. So you can just say let's look at fill color, right? So this is the default value, this is the mapping, right? And if it bypasses through to something, let's see what that means, right? So here I can say column fill color, I can say clustering coefficient, right? And obviously I can't have a pass through mapping here. If I have an integer, if color were an integer, right, if size, I can have a pass through mapping to degree or something like that. Higher the degree, higher the size. So here I basically make some sort of either a discrete or I can make a continuous mapping. Yeah. So see what happened to the figures.
Why can't size be associated to clustering coefficient?
No, size can be associated to clustering. See, here I am trying to color, right? So here node color, I can't map it to some integer. So it has to be mapped through in this continuous fashion. So I can't do a pass through mapping. There are three types of mapping you have in Cytoscape. There is a discrete mapping, a continuous mapping, and a pass through mapping. Pass through mapping is useful when you are trying to say node title. I mean, what is the label of a node? You pass through map the label of the node to the node ID or something like that. Right? Degree, again you can pass through map it, right? So the size is very small. Clustering coefficient will be a float. So you don't want to have, and all numbers are going to be between 0 and 1. Why would you want to make a pass through mapping of it? You make a discrete mapping, right? You will say 0.3. I'll just show you how that works. But you can now see this. See, you can. Maybe we should have a reverse mapping. Yeah. So lighter the node, higher the clustering coefficient. That's the mapping that's gone out currently. So you see all these dark nodes are basically 0 clustering coefficient, right? They don't have any triangles incident on them. So let's look at node size. So node size is a continuous mapping with degree. You can also have a discrete mapping if you want, which is actually painful. You can say degree value of 1, I will map it to what size, degree value of 2, I will map it to some size. If you have like just 5, suppose you have lethal nodes, non-lethal nodes, sick nodes, some 3-4 things like that, access via node attribute, you can map them to some 3-4 colors of your liking. So all these things are so trivially easily done with Cytoscape. Pass through is just pass through. So I can just pass through is just identity. So now I can do pass through mapping for So the size is 1 if the degree is 1, the size is 30 if the degree is 30. Pass through. Right. So 3 types of mapping: pass through, continuous, and discrete. You will rarely use pass through mapping almost, except for something like default it will be there. If you look at node label or something, as a pass through mapping for name. You can see that here. So it says that there is a pass through mapping for column name. You can also have other very interesting ways to do things here. So if your values aren't if your values are distributed in a particular way, let's just see. So let's say we're going to look at between a centrality, right? And I'm going to make a discrete continuous mapping, and this I can scale it the way I want, right? So minimum between a centrality is 0, wherein I'm going to have a size of 30, but after this I can say after a point I don't care, I'm just going to leave the same size. I want to really show the difference in size from this point to the next. So, anything beyond a particular cutoff, I am now going to make the size much different. Are you able to understand this? I can have more points if I want. I would not want to do something like this, but even something like this. This is almost like bringing it to a discrete mapping domain. So how does this look? I hope you got a quick introduction to Cytoscape in this video and also the very interesting database known as string, and I hope you now know how to load and visualize networks using Cytoscape and how you can visualize many of the network parameters which are computed using NetworkAnalyzer, such as say you want to size a node based on its degree and so on. All those things are easily doable in Cytoscape. In the next video, take a simple network and have a walkthrough of the different kinds of analyses one can do, and I will also mention to you briefly about other tools that exist.